everyone, welcome or welcome back to the channel, that is AMS MBBS lectures. In today's session we're going to discuss another topic from CVS Microbiology, and that is HIV and AIDS, and this is going to be the first part of this particular chapter. Now we have already covered the bacterial infections under CVS Microbiology. The videos are available on the channel, you can go and check it out if you haven't. And right now we're going to start with the viral infections under CVS Microbiology, and the topic that we're discussing is HIV and AIDS. As always, this topic is also divided under the following subtopics. So we're going to discuss the topic under introduction, structure of HIV, the HIV genes and antigens. This first three topics is what I'm going to discuss in this particular lecture. In the upcoming sessions, I will be covering up the remaining topics, guys. Okay? Then we're going to discuss the pathogenesis, the clinical diagnosis, laboratory diagnosis. Very important topic, that is NACO strategies for HIV diagnosis. With that, we have a look on the treatment and your post-exposure prophylaxis, and some other topics like the prognosis and monitoring of HIV, diagnosis of HIV in pediatric age group, and diagnosis of HIV in the window period, guys. Okay? Let's have a look on the introduction to this topic. When I say HIV and AIDS, it's very simple. HIV stands for the human immunodeficiency virus. Okay? If a person is getting an HIV infection, guys, please remember that the HIV infection is something that is going to occur in stages, guys. Okay? So if a person is having HIV infection, that does not imply that the person is having AIDS. Okay? Please remember that HIV occurs in stages, and AIDS is considered to be as the end stage. AIDS is considered to be as the end stage, the advanced end stage of HIV infection, guys. Okay? This is a very basic concept that I wanted to explain you all over here. Next thing, that when I say HIV, guys, this virus is a retrovirus. Okay, matlab retrovirus matlab what? It is a group of RNA virus. It's a retrovirus. HIV is nothing but a retrovirus, which is nothing but a group of RNA viruses. And please remember, these are characterized by the presence of an enzyme called as your reverse transcriptase, and this enzyme reverse transcriptase is the one which is going to help this viral RNA to get converted into the DNA, guys. Okay? And it is helpful in the pathogenesis of HIV. So while I will be describing the pathogenesis, I will explain you the role of reverse transcriptase in much more detail over there. Okay? Also, guys, this HIV, this virus belongs to the family Retroviridae, guys. Okay? It belongs to the family Retroviridae family ko belong karta hai. And also remember that this family Retroviridae is having many genera in okay, into which, of which only two genus are pathogenic to humans, guys. Okay? That is the genus lentivirus, which includes the HIV1 and HIV2. Then it also have the genus delta retrovirus, which is having the human T cell lymphotropic virus 1, that is HTLV1 is a part of genus delta retrovirus. The main concern of this particular topic is the genus lentivirus, which is including the HIV1 and the HIV2, guys. Okay? So that was all about the introduction to the topic, guys. Now let's have a look on the structure of HIV, guys. Okay? Structurally, just by looking at the image, it's very clear. What is the shape of the HIV virus? It is spherical in shape. Okay? It is spherical in shape. What is the size? The size ranges from 80 to 110 nanometers, guys. Okay? Now, please remember, the HIV virus is an enveloped virus. Okay? It is an enveloped virus, and it is having two components, that is the lipid part and we have the protein part. Lipid part is something that is derived from the host cell membrane. So the lipid part is made from the host cell membrane se derive ki jati hai lipid part of the HIV virus envelope. Okay? The protein part is made up of two components, and mainly it is the glycoprotein. Okay? Both the components are nothing but the glycoprotein. The two components are nothing but the glycoproteins, guys. Okay? It is made up of glycoproteins. Okay? And the two glycoproteins that are a part of the envelope, those are your GP120 and we have the GP41, guys. Okay? GP120, from the diagram it's very clear, these structures over here, this is the glycoprotein 120. Over here we can simply describe this GP120 as your knob like spikes on the surface. Okay? Knob like spike structures, okay, on the surface is what is called as the GP120. And over here the second component, that is the glycoprotein 41, it is going to form the it is forming the anchoring transmembrane pedicle, anchoring transmembrane pedicle. This is what it is forming over here. Okay. Next thing, guys, can you see this structure that I'm highlighting in the diagram? This structure over here, the one that I'm highlighting, this is what is called the nucleocapsid over here. Okay. Now this nucleocapsid is having an icosahedral symmetry. Okay, it is having an icosahedral symmetry. Okay, and it is made up of core proteins. Okay, it is made up of core protein से बना होता है nucleocapsid, and it is okay. It is enclosing two structures, that is the RNA as well as the enzyme. So it is having the RNA and it is also having some viral enzyme. Okay, please remember the HIV is having two identical RNAs. Okay, two identical RNA होते हैं, ठीक है, which is single stranded positive sense linear RNA, single stranded positive sense linear RNA होते हैं. How many? There are total 2 RNA inside a single HIV virus, guys. ठीक है. Viral enzyme, as I already said, HIV is a retrovirus, so it is definitely going to have the characteristic enzyme that is called as your reverse transcriptase enzyme. Okay, it is having the reverse transcriptase enzyme. Along with the reverse transcriptase enzyme, it is having an enzyme called as your integrase. Okay, integrase. It is also having an enzyme called as protease, guys. Okay, so that was all with respect to the structure of HIV, guys. Clear? Next thing, that is called as the genes and antigens related to HIV, guys. Okay, so there are total two types of genes. So we all are having 3 structural genes and we all are having 6 non-structural genes, also called as your regulatory genes. Okay, the 3 structural genes are the GAG genes, we have the Pol gene, and we also have the ENV gene. Okay, then when it comes to the non-structural regulatory genes, as I said, there are total 6 in number, which are further divided into 2 categories as your essential and accessory regulatory genes. Okay, so we are having 3 essential regulatory genes and 3 accessory regulatory genes. Okay, so essential genes are TAT gene, NEF gene, and the REV gene. Then we are having the VIF, VPR, and the VPU gene under the accessory regulatory genes. Let's have a look on the details of the structural genes. So here is a very simple table that is including all the information regarding to the structural genes. So when I say structural genes over here, it is simply implicating that these genes are responsible or are coding for the structural components of the HIV virus, guys. Okay, all these 3 genes are going to code for some or the other structural component of the HIV virus, and these genes, देखो कोई भी gene है ना, it will undergo transcription, translation, and will produce a protein. So all these structural genes that are shown, okay, that are written over here, they are usually expressed as a precursor protein, and this precursor protein is further cleaved into smaller components, and that is something that is going to, okay, form the structure of the HIV virus, guys. Okay, so that is the basic concept that I want to explain. So when it comes to the GAG gene, guys, okay, when I'm describing the GAG gene, it is coding for the core and the shell of the virus. Okay, the precursor protein that it is forming is the P55, and this P55 is cleaving into the P18, which is responsible for the matrix of the shell antigen, and the P24 and the P15 protein, which is going to form the core antigens को बनाते हैं ये. Okay, then we similarly we have the Pol gene over here, the POL gene, which is responsible for the coding for the viral enzymes, that is your reverse transcriptase, protease, and the integrase. So viral enzymes के लिए we have the POL gene. For the core and shell we have the GAG gene, guys. ठीक है, Pol gene is the one, उसका जो precursor protein होता है, it is cleaving as P31. P31 is responsible for the integrase. ठीक है, then P51 is something that is your reverse transcriptase, and we also have a P66 component of the Pol gene, guys. Okay, similarly we have the ENV gene. From the name it is clear, ENV, it is coding for the envelope. Okay, envelope का भी glycoprotein को ये कहीं ना कहीं करता है, not the lipid part. Why not the lipid part? Because lipid part of the HIV virus is something that is derived from the host cell membrane. So ENV gene, the envelope gene over here, is going to code for the envelope's glycoprotein. And when I say glycoprotein, abhi structure mein dekha tha, that is GP120 and GP41. Please remember that it is the precursor protein is GP160. 160 is the precursor protein which will cleave into something called as GP120 and GP41. GP40, okay, GP120, this glycoprotein 120, it is the main receptor of HIV, and GP41 is the fusion protein. Now, what is this main receptor? What is this fusion protein? So, okay, see, right now, just remember that it is the ENV gene. Uska precursor protein GP160 hai, which cleaves into GP120, which is the main receptor for HIV, and GP41, which is the fusion protein. Okay? Details of what is this main receptor, what is this fusion protein, you are going to understand this thing under pathogenesis of HIV. Right now, just remember the details of the gene. Understood? Moving on ahead towards the next topic, and that is so that was all about the structural genes, guys. Okay? So we have covered a lot of topic, that is your introduction, the structure of HIV, as including the HIV genes and antigens. So the first three topics are something that we are done. Right, okay, now that is it for this session, guys. In the upcoming session, I would be covering up the pathogenesis and clinical diagnosis, and the subsequent topics would be covered up. Okay? So that is it for this session. If you found this content useful, then do like, share, and subscribe to the channel, which is AMS MBBS Lectures. See you all in the next one. If you have any questions or queries, comment down. Bye bye.